Good afternoon and welcome to today's webinar on revolutionizing prospective studies and registries with automation and AI. To reduce background noise, the lines will be muted during the presentation. If you have a question during or at the end of the presentation, please enter it in the question and answer box located in your webinar control panel. Presenting today's webinar is Dr. Richard Gliklich. Dr. Gliklich is the CEO of OM1. Previously, he was president of the Outcome Division of Quintiles and the founder, CEO, and chairman of Outcome. Dr. Gliklich is the senior editor of the U.S. Agency for Healthcare Research and Quality Handbook, Registries for Evaluating Patient Outcomes, a User's Guide, and the PI for the Outcomes Measures Framework. He holds several patents for both health outcome systems and medical devices. Dr. Gliklich is a graduate of Yale University and Harvard Medical School, a former Charles A. Dana Scholar at the University of Pennsylvania, and a surgeon at Mass Eye and Ear Infirmary, and the Leffenfeld Professor at Harvard Medical School. Thank you for presenting today, and I'll now turn it over to you.
Hi, my name is Rich Gliklich, and welcome to done is we've changed the paradigm of clinical research from recording data on paper case report forms to recording data on electronic case report forms. But still, most of that effort is largely manual. And that's associated with significant site burden, high costs, long timelines, low site satisfaction, and potential for biased enrollment. And I say potential for biased enrollment because when the workload is high, clinicians are going to look for times during their day when the workload is less to be able to enroll patients and suggest clinical studies to them because of the amount of manual data collection that's required. And yet, most data in the US healthcare system is digitally available and has been digitally available at least since 2014, 2015. In fact, as of 2021, 78% of office-based physicians and 96% of non-federal acute care hospitals had adopted a certified EHR. And we hear about certain technical solutions which enable the collection or movement of data from the electronic medical record into the electronic data capture systems, but those technical solutions must be lacking because little has changed. We could talk a little bit perhaps during the discussion about why that is: access to ancillary documents, ability to process unstructured information. But in some ways, the technology is not quite right, or perhaps the regulations are not quite clear, or maybe there just has not been enough benefit to move the needle. I want to talk about two concepts. On the left is traditional data collection, or what I'll call traditional data collection, and on the right, automated data collection. Traditional data collection is everything that we're used to. We screen and consent patients. We enroll them. We randomize them to particular treatments or to controls. Baseline visits get entered. Patients return for follow-up visits, whether in person or remotely, and those assessments get entered into case report forms. We obtain images, pathologies, laboratories, and we transcribe the results into the electronic case report forms. That is the nature of the clinical trial enterprise. Automated data collection, the term I'll use, really refers to using data automation to leverage electronic health record retrieval, including notes from the encounters, procedures, laboratories, pharmacy systems, perhaps ancillary systems like laboratory information management systems or radiology image management systems, bringing in images, pathology, tumor registries, and now wearables. Essentially, what we can pull from the electronic health record and other systems is almost everything except for specific clinical outcomes assessments, or ECOAs, that we want the clinician to obtain, or specific patient-reported outcomes that we want the patient to report to us, as well as additional assessments that are not captured in routine care. In addition to being able to bring data in from sites or clinical centers or health systems, which is something that we're doing continuously from dozens of centers, there's also the ability to obtain individual consent and then retrieve their electronic health record from many different centers for each patient. That's particularly valuable around rare disease and now also possible. And then the last area that we would consider automated data collection is passive data in the form of linkage to medical and pharmacy claims data, social determinants of health data, death or mortality data, and so forth. These are data that we can bring in much more passively. So what does that mean? It means that traditional data collection is bogged down by labor. It takes work to do all of the things that we see on the left. Whereas automated data collection is leveraging electronic or digital information in the way it was meant to be leveraged, by harmonizing it and transferring it into clinical systems so that it doesn't have to be repeated in terms of capture. The benefit there is no labor. As you can see, what I changed on top is really, I don't see it as traditional versus automated data collection. I think the future is about traditional data collection augmented with our ability to use automated data collection wherever possible. And the good news is that regulators are not just leaning in, they probably have been ahead of this movement for some time. I'm just showing some of the various guidance documents or handbooks that are used for understanding what is appropriate or acceptable in determining fit for purpose data that might be submitted to a regulator. And you see the regulations or the guidance documents on use of electronic health record data and clinical investigations, considerations for use of real world data from medical claims, from EHRs, from other systems of record. And one comment by Dr. El-Zarad from the Office of Medical Policy earlier this year is that the use of AI, including to facilitate data collection, are really transforming the ways that drugs are developed and used. And to me, data automation is the future of clinical research. So here, I've changed the naming. Instead of calling it traditional data collection and automated, I'm calling it active and passive. So passive data collection is that which we can collect without requiring work effort by a clinician, by a research assistant, by the patient. And that includes those direct consented EHR retrievals, health system center EHRs and ancillary systems of record, and then linked data from commercial and federal insurers, from pharmacies, from mortality, social determinants of health, from third-party laboratory systems, and the list goes on. Whereas active data collection is just that which we cannot otherwise collect passively. Of course, we need to obtain consent from the patient. We need to screen them. We're going to randomize them. When we enroll them, we need to do that. But then up for grabs is whether or not the baseline assessments are needed beyond specialized clinical laboratory assessments in ePro, and whether prospective follow-up visits are needed and what types are needed and how much is actually required because it's not already contained within these passive data sources. So much so that we have one study that's currently running where we reach the point of randomization and everything is otherwise collected passively because standard of care already collects the key data points. That's not usual, it's not even typical, but we're moving more and more towards a world where more data can be accessed passively, requiring less active data collection and more focus on the active data collection, producing data sets that are more robust, have higher site satisfaction, ideally lower costs, and can be submitted for regulatory purposes. What enables all this? So I'm going to spend the rest of the time talking about the fact that the technology is here, it's ready, it's transformative, and it's powered by generative AI. This is a diagram of what AI-powered data automation looks for us. This is our system or system of platforms called OM1 Aspen. And what you see is that we connect to electronic medical records and other systems of record on the left and start bringing data through a series of steps or microservices in a pipeline that ultimately delivers the data into an electronic data capture system to be linked with other data under PI signature or to other databases. What I've shown here is in orange the steps in that transformation pathway that are done by generative AI. And I'll talk about some of them because it's both novel and exciting. And then on the bottom, what you can see is that in this type of pathway, data can be utilized for regulatory purposes because traceability is maintained, it's auditable, and we can obtain electronic signatures. So starting from the left, we bring in data initially from the electronic medical record, and we connect and ingest that. Of course, data is quality controlled and brought through. One of the steps we can then take is to tokenize the data. That means allowing the data to have a token generated that can allow it to be connected to other passive data sources like medical claims, pharmacy claims, third-party laboratory data in the future. Not always required, but a nice to have. We could also choose to de-identify the data for certain types of studies. If we're running a retrospective study and we want to perform it under a waiver of informed consent, this is often a requirement by the IRB, and that step of de-identification is also the step of tokenization, but we'll also use AI in this case to de-identify the unstructured record or the clinical narrative, procedure notes, laboratory reports, and so forth. The next step in that process is to create adapters or connectors between the electronic health record system, which is in a certain data model, into the common data model that we hope to have our research data in. And that is increasingly being performed with the help of generative AI. Another step is to extract data from clinical notes. That's leveraging unstructured data, and I'll show you an example of that, as well as extracting data from the metadata around images, DICOM images, where we're going to pull in report information or other specific information from those images. The fifth step is to enrich that data. And by that, I mean enable there to be more outcomes, because ultimately what we want to understand from clinical information is endpoints and outcomes. And now there's a variety of techniques to enable us to do more of that, again, leveraging generative AI. Sixth step, which is incredibly interesting and useful, particularly in subtyping diseases or in rare diseases, is we leverage phenotypes or a phenotyping engine. And I'll go into details on that. And then there may be other steps where from a large data set, we create different cohorts. We may link additional data. We may want to explore the data as it's being processed and leverage generative AI for that as well. And I'll go through some examples as we continue. So let's talk about this process of extracting data from notes and images, because it's relatively new, meaning over the past 4 or 5 years. It's highly accurate and it's highly reproducible. So this is an example where we're leveraging medical language processing. Now, there are different ways to pull unstructured information into structured data for research purposes. We can hire a team of abstractors. We can use technology to enable those abstractors to focus in just on the notes or the components of the record where the information we want to extract is likely present. Or we can turn to the system to process the language using things like LLMs to bring in specific data and then use our abstractors to validate that what the algorithms we've used are valid and reliable. And that's the case here. So here what the challenge is to identify the location of the dermatology problem, in this case psoriasis. And you can see how we're able to process that to identify, differentiate between elbows, flanks, and the lower back in this example, as well as identifying the date for that extraction. What's really interesting is that this capability not only performs at about the same level as abstractors in terms of accuracy, and maybe even a little better, but the recall or the ability to find the information in and among thousands of charts is better using the machine. So we get more data points when we leverage the machine, even than when we leverage abstractors. Another technology that we have been advocating for several years is endpoint amplification. And what this means is we take the unstructured note or the clinical narrative, what the clinician reports to the medical record in straight text and dictation, and from that, we're able to extract, using a version of sentiment analysis, specific endpoints that are outcomes. So in a condition like lupus, where SLEDAI is only observed in standard of care practice, a handful % of the time, as long as we can have a certain number of SLEDAI to validate our extractions, we can utilize any relatively complete note completed by a physician to identify an estimated SLEDAI score. And we have validated that and published on it, as we have for many other amplification models, such as in MS, looking at EDSS, in rheumatoid arthritis, looking at CDAI, in heart failure, looking at New York Heart Association classification, and depression, looking at PHQ-9. And what you see is not only do we get more endpoints, we get more patients with endpoints. And the increases can be very, very dramatic, as you see here. Why is that valuable? So let's look at an example of a patient with an estimated endpoint, just looking at one specific patient with lupus. And what we see are the blue X's and a dotted blue line that reflects what was observed in the medical record. So these are the actual SLEDAI scores reported by the clinician, observed and recorded by the clinician in the medical record. And then the orange ones are the ones that we're able to actually estimate using the extraction algorithm. What you see is we have more data points for the specific patient over this interval. But the other thing, we see a different story. Looking only at the blue scores, what we see is the patient appears to be relatively stable with moderate disease activity scores throughout this time period, because 2 points make a line, 3 points certainly make a line. But when we look at the orange, we see that's only partially true. So in July, we actually see that the patient's scores have improved to mild. And then following July of 2018, they start worsening and go up as far as high. And what we learned is that in July of 2018, the patient had been doing so well, their disease-modifying therapy was discontinued. And so what you learn is that there's missing information always, and observed information coming from the electronic medical record, but we can fill in gaps using this type of technology that's incredibly exciting and powerful. I want to talk about the concept of phenotyping. You're all familiar with the concept of genotyping. So if I have a cheek swab, I can put that through an Illumina array, and it will look for 500 or more single nucleotide polymorphisms, or SNPs, associated with different gene defects. And then we'll group those gene defects, and we can analyze them with respect to genotypic signatures that might relate to a patient's having a particular disease or risk of the disease in the future. Similarly, with the medical record, we can do this digitally using the concept of phenotyping and this platform technology called PhenOM. So what we do with PhenOM is exactly what we would do with a gene sequencer. We're now looking at the digital record in a 500-dimensional space, things like disease severity, things like evidence of progression or prior treatment history, comorbid burden, the effects of mental health or social determinants. We can also even include physician experience in that. And from that, we're able to similarly identify areas of this fingerprint that get painted that we can now associate with specific undiagnosed diagnoses, or the likelihood of a particular patient to respond to a particular treatment. So how do we utilize that technology? Well, I'm going to give you a couple of examples. One is to create a patient registry with an uncoded condition. So in this case, the uncoded condition is treatment-resistant depression. Treatment-resistant depression has no strict definition or standard diagnosing code, making it detection difficult. But what we've done is we created a phenome model, if you will, that identifies patients with a high likelihood of having treatment-resistant depression and validates that against the clinician actually recording that the patient has treatment-resistant depression. We've also done it versus the regulatory definition of treatment-resistant depression. But this model is looking at the physician actually determining the patient has that. And now we can apply that technology against any digital record with an area under the curve here of 0.92 to identify patients just based on the coded information that are likely to have treatment-resistant depression, and then be able to follow them longitudinally, retrospectively, or prospectively through electronic medical records and other source documents as a patient registry in an uncoded condition. Another use case, which we're utilizing in rare diseases, is really also paradigm shifting. In this case, what we're leveraging is that same platform that I showed you before, Aspen, where we're connecting and ingesting data, we're QCing it, we're mapping it from health systems, but now we move up the identification of phenotypes. By moving up the identification of phenotypes, we can determine electronically whether or not a patient meets eligibility, inclusion-exclusion criteria, and may have an undiagnosed condition that may be relevant to a trial or to treatment. And then that patient can then move on to confirmatory tests. So for example, in the case of something like Fabry disease, we can then refer that patient for enzymatic testing and potentially for gene testing to confirm what the system has identified as a likely patient with Fabry disease. The next thing I want to talk about is very, very different. This is a different approach to leveraging generative AI in a research platform like we've been discussing. But the purpose here is to leverage it for analytics. Now, typically analytics are they're the province of the epidemiologist and the statistician, and that is all well and good. But you have more business users or naive users or clinical users who would like to benefit from being able to explore the data that's being collected in a more chat-based interfaces. So that's what we've now developed, this OM1 Data Exploration tool, which allows the user to interact with the data that's been collected either for a specific research study or across all of our data set, 350 million people, straight through a chat interface. And I'll show you just quickly an example of that, where here what's happening is I'm writing in a query to find patients who have a diagnosis of psoriatic arthritis. And I've been through this enough to know that we also want to indicate how that diagnosis is made. Don't have to do that, but I've chosen to do that here, saying that they have at least a pair of diagnoses at least 30 days apart. And in this case, I'm looking for safety signal associated with the prescription of methotrexate during a defined period. And what I'm asking the system to do, just based on my query going against the database, is to give me a count of the number of patients who fit the diagnosis that I'm asking for and who receive methotrexate, and the number who are hospitalized in the 12 months following the first prescription. So let's follow that through. So what happens here is that the query is submitted, and what the system will do is now determine the logical steps of that query. It will disambiguate those steps if it finds that there's ambiguity in how the step is done. And it's generated SQL on the right that it runs against the database, and then answers on the left. So here you see that the SQL is being generated on the right, and then the answer to the question is we have about 7,000 patients prescribed methotrexate between January 1, 2020 and December 31, 2022. And about 400 of them were hospitalized within 12 months after their first methotrexate prescription date. Now that is not telling you a safety event, but it's telling us where to look for that safety event. We can now take this further use chat to refine the cohort, further use chat actually to ask the system to go as far as to generate statistics that we can even use in an abstract for submission to a clinical meeting. That is the state of interfacing with data today, and it's super exciting, particularly for those of us who are not as good with statistics. I want to move on to something that we feel is really important around using these capabilities, particularly around active data collection and passive data collection, and that's design. So what we want to do when we design a program, whether it's a clinical study or a registry, so we want to design it with respect to its major purposes, choose the study design, select the right data sources and sites, identify the target population, determine how that study population should be recruited, look for bias and so forth. And to help our customers work with this paradigm of active and passive data collection, we've created a center of excellence with people with tremendous experience specifically around this, because we think it's really important to both limit active data collection, but to collect the right active data, such as clinical reported outcomes and patient reported outcomes, and to maximize passive data sources from these different sources in the right format, including understanding how to ensure traceability and auditability for FDA and other regulatory inspections. Let's walk through a couple of case examples, and maybe a handful of them, of using these technologies to automate data collection, leveraging automated EMR data collection, and occasionally some active data collection. So this is a case where we're looking at a relatively rare disease, cystic fibrosis, in about 15 centers over 2 years. And the real purpose of using automation in this case was not the number of patients, but the leveraging of the unstructured data in combination with the patient-reported outcomes data to actually compare how clinicians reported patient success with this new therapy versus how patients were reporting it. Very different and much larger study is a program that we've undertaken for a large-scale comparative study for a cancer product for its label expansion. In this case, we're leveraging data from over 600,000 patients, in this case collected retrospectively for a product that was in use, but seeking a new label indication from about 10 sites. And what is most interesting, I think, is the site NPS score. What is the site NPS score telling us? Well, the Net Promoter Score for sites is really telling us how happy the sites were with the program. For those of you familiar with a Net Promoter Score, a score of 20 or above is considered pretty good. When you get above 70, it's incredibly good. So what we see with this automation, even though we have hundreds, literally hundreds of thousands of patients in a regulatory grade study that's having data submitted to a regulator, super high level of satisfaction from the sites. Using the same technology, we've looked at creating a large registry, in this case, over 1 million women, both retrospective and prospectively, to follow them over time. These are all women who qualify for screening for breast cancer. They don't necessarily have breast cancer, nor do they necessarily have screening, but they simply qualify for screening. And by that, we can then begin to look at questions not only of results, we can even look at questions as who gets referred for screening and who does not. And then are there differences by things like, in this case, race, ethnicity, access, and so forth. So again, creating very large cohorts to be able to study incredibly important questions, and increasingly being able to also look at things like bias in design and treatment access. Very different use case is this, which is a reusable real-world evidence and patient experience platform that also allows for provider interaction. So here we're combining, again, passive data collection from the electronic medical record on about 10,000 patients for about 10 sites in a neurological condition, with patient-reported outcomes and actually diary information, combined with the clinician's ability to view the same portal as the patients, and in fact creating a community. Another example is around post-marketing commitments. So again, post-marketing commitments may have very high patient numbers. When we get to very high patient numbers, it's all about scale and being able to perform these studies at a reasonable cost. Otherwise, it can take many, many years to complete them, or it can be extremely costly. In this case, we're looking at 50,000 patients where the post-marketing commitment provided a new label indication, but required that this commitment be undertaken. And we're, again, performing it using an automated approach where we're going prospectively in each quarter, pulling data from these centers, processing that, both structured and unstructured data, to yield the results. In this case, again, the site NPS score is recorded and is way in excess of 20. It's around 71. I'll show you one last example before we talk about some other things. So this is a study, and I alluded to it earlier, where what we really wanted to do was identify patients for the purpose of further confirming a diagnosis. So what we did here is in major medical centers, we applied a predictive model based on PhenOM, or that phenotyping technology, which has an area under the curve of 0.82. And what we do is apply it against the entire database of patients of the health system, identify ones in the higher echelon of risk for the underlying disease, and then have those patients enrolled in a study which confirms the presence of the disease through enzymatic and genotype testing. It is a much more efficient way to identify patients for disease with a rare disease, both for research purposes and treatment. It creates a cadre of patients that then can be followed in a registry longitudinally. And again, it uncovers a disease for which patients may not know that they've had it, even though they have a number of symptoms or patterns of care that would have suggested it to a computer, maybe not to the rest of us. I want to touch now on something that's really near and dear to all of us, particularly in these, a little bit more economic challenging times, is the cost efficiency of data automation. What I've plotted here on the x-axis is the number of subjects per study, and on the y-axis, going vertically, is the cost in dollars per patient per study. And what you can see is that as we increase the number of patients using data automation, the cost curve improves exponentially, and it improves exponentially early on. So what we're seeing is that the technology scales incredibly well, that even at lower numbers of patients, at the 2,000 patient level, the costs are highly competitive and may be better than traditional means. And then as we scale up these studies, the costs drop dramatically, so that when we exceed 50,000 or 100,000 patients, we're getting into tens of dollars, not even hundreds of dollars, to run studies like this. So it's incredibly compelling what this type of infrastructure can do from a cost perspective. So what are the use cases that we are seeing and implementing for this type of program? So one, I've already talked about it, disease and product registries. Registries, we really want to enroll patients by an exposure to a disease or exposure to a product. And so that can be done essentially through a query, and then we can follow those patients passively, other than the very specific active data we may be seeking, like clinical outcomes assessment or PROs. There's a lot of overlap between those types of studies and non-interventional and observational studies or phase 4 studies. I talked about post-marketing commitments. We talked about ECO and ePro studies, which is the active component, but we want to pair that with the clinical data from the EMR. This technology is robust for external control and comparator arms to link to a clinical trial. It's robust for trials that you want to embed in a registry, where whether you have a rare disease or a chronic disease, where the infrastructure for collecting data in an automated way is put in place around a registry or a cohort study. And then trials can be embedded in that at a very much lower cost per patient, because the underlying registry and infrastructure both helps identify patients for the trial or study, and then allows the following collection of the passive data for these patients. It's also true for large simple trials or pragmatic trials, where the only difference between the control group and the and the test group or the experimental group might be the randomization, and all other data might be collected similarly. Also, this concept enables us to consider leveraging passive data collection in any clinical trial. Every clinical trial, in my view, particularly phase 3 study, should have the opportunity to collect long-term patient follow-up based on real-world data on patients. And this technology enables you to do that in a very cost-effective way. We're using this capability around quality measurement programs for healthcare organizations, identifying patients who are not being treated to guidelines and changing that. And then we're also enabling clients to use individual microservices. So what you saw in that pipeline, we can expose that as microservices. So we can receive text, digital text, and return back extracted information through what we call auto curation. We can receive back a clinical note in a number of different condition areas and return back an estimated endpoint of the types that I showed you. We can receive information in a standard form against an API and return back a digital phenotype, a subtype, or that can be used for treatment or research or prediction. And then, one of the more exciting things is when utilizing our model, our data model, any end user can interact with the data set in a chat interface that's continuously getting better. We now have clients who we're leveraging as pilots to use that and help us improve the capabilities across different use cases. One comment or opinion I wanted to put out there based on what I've been talking about, and I own this comment, is that with data automation and registry infrastructure, I believe the research paradigm changes. And I alluded to this before. I think that now we're at the point where cost effectively every clinical trial, in particular phase 3, phase 3B, has the opportunity for a low cost, real world, long-term follow-up study or registry. That patients can be consented for that upfront, can be followed with the ability to have real world data over a long period of time for any number of different purposes. And I also believe every clinical trial benefits from the reduced cost and improved time to data that this data automation infrastructure enables. And that's why I believe that this is going to be paradigm shifting over the next few years. First, the move to leverage the technology to bring EMR data into EDC systems or directly into databases in a much more cost-effective, scalable, and accurate way than what has been available before. And then to leverage these other GenAI capabilities to truly automate data collection for clinical trials and registries. So again, it comes from this type of AI-powered data automation, the technologies that I've been reviewing with you, and the ability to do this in a regulatory compliant way. Takeaways I hope you bring from this discussion are that prospective studies and registries are too manual, costly, and burdensome, and they don't need to be. They've been that way since the 1990s, and the technology today is better than that. And that automation changes the paradigm, enables us to have passive data collection, limit active data collection to just what's needed, improve site satisfaction, dramatically reduce the cost of these studies and registries. We leverage linked data to fill in the gaps. Were patients seen or followed at a different site or center that we don't have the data to access to? Do we want to understand something about these patients, whether it's about their race, ethnicity, mortality, et cetera, that would best be filled by linked data? And as I've showed you, AI makes this possible, and it's extremely enlightening to see that. And regulatory guidance, I believe, particularly from the FDA, has enlightened the path. And I look forward to seeing further guidance documents that will help continue to help us all advance this paradigm. So with that, I'm going to go to questions. Thank you very much for listening to me. And
Great, thank you, Rich. As a reminder to our audience, if you have a question, please enter it in the question box located in your webinar control panel. Some questions came in during the presentation, so we'll start with those. So given the number of studies and registries that OM1 has done, do you have an existing network of sites that we could tap into for our studies? In other words, how many health systems have you connected to in the past?
Yeah, great question. So we have connected to about 5 or 6 dozen centers, and each time we connect to a center, by center I mean a major healthcare system that has an average of at least 12 hospitals associated with it. So these are big systems that are pretty representative in total of the country, and we've created adapters to those EMRs. So if we're doing another program or study, it's a relatively shorter hop to gain that normalization that I showed you. Now, when you have a specific disease or a specific center of excellence around a condition area, there may be new centers that we bring on, and we're really good at bringing new centers into the fold, into this data automation. It's what we have a large team that's focused exclusively on that. So both are true.
Great, thank you. In describing the automated systems, as you mentioned that some of the capabilities are available as microservices, what does that mean in practice for our research?
Yeah, so we have clients who, for example, bring in their own medical records and have clinical notes and really just want to tap into or work with a site that is going to deliver them clinical information and want to extract from the note either text using that component or estimations. And we just set up what's called an API, which just means that the data hits our server and returns back whatever that packet of information, whether that's a set of symptoms from a clinical note on nasal polyps or a estimated PHQ-9 from a psychiatry note seeing a patient. So that's one set group of microservices. PhenOM, or the phenotyping, is also set up that way. So there's groups that want to leverage that either within existing systems or some against an electronic medical record system they might be partnering with. That just again can hit that microservice and return back the information. So the way to think of it is that the platform that we show you is a collection of microservices that are connected with automation, and components of that can be exposed or used by other systems, or you could just use part of the chain, if you will.
Great. Do you think AI could be used to identify patients for recruitment and help optimize protocols for further efficiency and cost savings?
Oh yeah, I mean, so a couple of ways that we think about it. Obviously there's something called a computable phenotype, which is really and that's a term of art that's used by the FDA and NIH, and that just means developing a computer algorithm which identifies certain inclusion-exclusion criteria. But what you can do with AI is not only that, you can also use the AI for its fuzzy logic. And when we think about clinical trial recruitment in, for example, our program called Polaris, what we do there is think about, if I am we're trying to ask the system to find patients like these, right? Find patients like these, show them some patients, find patients like these. What does that mean? Well, if I have a clinical trial that has patients required with a certain laboratory, but that laboratory is not being accessed in real world, it's just not something clinicians obtain, but AI can identify patients who are likely to have all those other inclusion criteria and probably have an elevated laboratory test. That's a good target to use AI to then bring that patient in, just do the laboratory test, see if they meet that last inclusion criteria, as opposed to just trying to find patients who randomly had all of those tests that you need to use. So that's one way we leverage AI. You can similarly leverage it against existing data where you want to identify or plan a protocol and run a protocol against existing data. But sometimes there's events, say safety events, that you would want to use a more fuzzy AI to identify the phenotype of patients who are going to have those safety events. So those are a couple of examples.
Okay, thank you. Can you describe a bit more about what the FDA is looking for in being submitted for RWE purposes from these types of programs?
Yeah, and I mean, I mentioned this as I was presenting. I think the FDA has done a really good job of laying down the breadcrumbs as to what they're looking for with RWE submission. But what you really learn is during the process of submission, how both astute they are and how much learning they're going through. So what they're looking for from the RWE perspective is data that they call fit for purpose, which means it has to go through a number of tests on missingness, which is a big problem in real world data, on representativeness, on potential areas of bias to meet goals. And then if you accomplish that, the last thing that you need is two things I mentioned earlier. One is traceability. You need to be able to go back to the source and be able to follow every transformation that happens to the data, and auditability. And that means that the FDA needs to be able to send an inspector to the clinical site where the EMR is, where the data originated, be able to look at that screen, compare it to what they see in this, what the doctor would see in the screen to what's in the database. That's what we call auditability. So as long as you hit those things and you engage in conversation early and often with the FDA, I think you're on a good path to meet both fit for purpose and usability for data in real world evidence studies with the FDA.
Okay, we have one more question here. Are there additional costs when a partner wants regulatory grade compared with research grade?
So there are additional hurdles that are necessary to meet those traceability and auditability components of the data. I wouldn't say they're massive, but they are checkboxes that you have to hit. I'll just give you one example. If we're utilizing data from say one of these programs and we want to just use it for publication, all of the things that we typically do, IRB approval, getting the sites on board, et cetera, getting the data, processing the data in this automated way, are there. But if a site is going to be audited by the FDA, they need to know that in advance and agree that they're going to do that, and also maintain a certain level of documentation within the site itself about how they handle the data, whatever that might be, before it was received by us. So there is an additional layer that has to be considered and utilized. And there's some other things as well that would be best practice. Is it hard and fast? It's not hard and fast. Again, the FDA uses this fit for purpose case, and it depends what the purpose is, and maybe certain things get relaxed. Maybe you're not going to have a BIMO audit. Maybe these other things won't occur, but that's where the additional costs can come in around planned regulatory submission versus not regulatory submission.
Great, thank you. If your question was not answered or you have one following the webinar, please submit to us directly and we'll follow up with you. Thank you again for joining today's webinar. A link to the recording will be distributed to all attendees, and we hope you'll join us again in the future. Thank you.
Thank you.